Good evening, dreamers. Welcome to Dreamtime Science, the place where wonder slows to a whisper and curious minds drift into sleep. If you've arrived here, you're probably searching for something equal parts soothing and slightly nerdy to carry you toward tomorrow's sunrise. You're in the right place. As we nestle into the soft embrace of the night, let's prepare to journey into the infinite spirals of our inner universe, the mysterious world of DNA. Like secret messages whispered in the hushed tones of slumber, our genes carry the blueprints of our very existence. Let's drift with a gentle, dreamlike curiosity into this intricate dance of life's instructions. Before we begin, may I ask a tiny favor? If this kind of midnight science story helps you doze, a quiet like or subscribe lets this sleepy corner of the internet keep glowing. And because I love to see how far the starlight travels, where are you listening from and what time is it there? Slip your answer into the comments below. I read them like bedtime postcards. Now take a moment, adjust your blanket, find the perfect hollow in your pillow, let your shoulders melt, breathe in and out. Ready? Then close your eyes and let's wander together. As the soft candlelight flickers gently amidst the quiet shelves of this ancient library, let us resume our leisurely journey into the realm of life's quietest architects and deepest whispers. We have wandered through the vast expanse of cells, marveled at the delicate dance of molecules, and now, step by step, we draw closer to the heart of the tale of DNA, the blueprint of life. Imagine, dear listener, that somewhere hidden within every living thing, from the tiniest seedling to the majestic oak, from the gentle flutter of butterfly wings to the slow, measured pulse of a sleeping whale, there lies a carefully folded scroll, long and intricate, filled with instructions older than the mountains, and yet ever new with each breath. This scroll is DNA, a strand so delicate, so masterfully coiled, it contains the masterpiece of design, the blueprint that sketches every creature's form, function, and future. Let us picture now, very softly, the silver moonlight spilling through a high window onto a desk scattered with ancient parchments and faintly glowing manuscripts. Among them lies the DNA, a double helix, like two winding staircases entwined in an eternal embrace. This spiral staircase is composed of millions, billions perhaps, of tiny steps, each step made from a pair of letters, 4 in all, A, T, C, and G. These letters, though simple, form the language of life, the code by which every living cell understands what it must become and how it must behave. Long, long ago, before the dawn of human memory, when the earth was still young and the first whispers of life stirred in the ancient seas, these letters began their quiet conversation. They arranged themselves in sequences, patterns that formed the earliest instructions, and so began a remarkable story of survival, change, and complexity. Each sequence folded into itself a secret song, a recipe for proteins, the tireless workers and architects inside cells who bring this story to life. Now, inhale slowly and imagine wandering through a vast forest of chromosomes, these great towers standing tall within each cell's nucleus. Inside those towers, amidst millions of tightly packed DNA threads, are the chapters of life's book. Chromosomes are the neat bundles, the carefully organized libraries protecting and preserving this precious genetic script through countless generations. As we linger in this silent forest, notice that every living being carries these chromosomes in unique bouquets. In humans, there are 46 of them, paired in a delicate balance between the traits we inherit from our mother and those from our father. These pairs gently unfold around the DNA, each spool carefully wrapped for safekeeping, like ancient scrolls in velvet-lined cases. And what does this script say? It encodes for the molecules that build the body, for the enzymes that manage the chemistry of life, and for the signals that guide intricate development. Each gene, a phrase within the grand narrative, holds the information for a single protein. But it is not mere letters. It is a story of beginning and continuation, of replication and expression that quietly whispers, "Here is how to live." Let your thoughts drift now, like mist across a still pond, to the process of reading this script. Within each cell lies the mechanism of transcription, a gentle, methodical reading aloud, where a messenger is sent forth from the tightly guarded nucleus into the bustling cytoplasm. This messenger, a molecule of RNA, carries a copy of the genetic instructions, like a scribe copying from a precious manuscript, to pass along the tale to eager builders and makers within the cell. While this copy rests transiently, the mighty ribosomes gather, like attentive listeners awaiting a story. Through the elegant process of translation, they read the RNA's message, weaving amino acids, the building blocks of proteins, into chains that fold and twist into living structures. It is a slow, deliberate craft, a weaving of life's fabric thread by thread. Now, as your breathing deepens and the quiet rhythm of night embraces you, picture the DNA's twin spirals moving gently, unzipping their bonds like two strands uncoiling to reveal their secrets. This unzipping is the first step in replication, the process by which life ensures its own continuity. As one strand opens, new nucleotides, those chemical letters, mirror their partners, creating a precise copy, a twin scroll that will journey on to the next generation of cells. Cells divide and reproduce, and with each cycle, this process must be flawless, for errors or mutations can subtly alter the script. Yet this very imperfection is also the wellspring of evolution, the seed for diversity, adaptation, and the endless variety of life forms that roam, swim, and fly upon this earth. Imagine, then, the boundless beauty hidden in this delicate balance. On one hand, the steadfast preservation of ancient instructions. On the other, the gentle allowance for change, for new verses to be written in the book of life. Flow with this imagery, moving deeper into the quiet understanding that within each living being is a library of blueprints so vast it defies imagination. Each cell, a keeper of knowledge passed down through eons, holds within it the promise of tomorrow and the whisper of ancestors long gone. And as this truth settles softly within your mind, think now on the wondrous history of discovery that brought this intimate knowledge into human hands. In the 1950s, in a humble laboratory, two scientists named Watson and Crick glimpsed the twisted ladder of DNA and uncovered its form, a structure that mirrored the infinite beauty found in nature's design. This revelation was not abrupt. It was the culmination of countless moments, quiet experiments and gentle deductions, converging to reveal how life stores its blueprint so elegantly in a language so remarkably simple yet infinitely profound. Their model showed at last how the sequence of these four letters, adenine pairing with thymine and cytosine with guanine, form the rungs of the ladder, allowing it to replicate, to be read, and to guide. Such knowledge has since unfolded new vistas, helping us to understand hereditary diseases, the diversity of species, and even the mysterious connections linking all living things. Let the calm of this discovery seep into your thoughts. Imagine yourself there in that quiet lab, the air thick with anticipation, the turning of pages, the careful measurements, and then the moment of clarity, like the first star emerging in twilight. Here the mystery of life began to reveal itself not as a distant enigma, but as a beautifully ordered dance, written in a code both intimate and universal. As you sink deeper into this serene reverie, visualize the molecules themselves, their elegant shapes, the hydrogen bonds forming and breaking, the helices twisting into the night, dancing softly in the dim light of the cell's nucleus. Each atom, each bond, working tirelessly in perfect harmony, unfolding the instructions that guide the miracle of existence. Drifting ever deeper, allow your mind to rest upon the immense power contained in such a tiny structure. DNA, a thread only 2 nanometers wide, holds the sum of life's knowledge, a vast encyclopedia compressed within a spiral of molecules. It is a code read millions of times within your body each day, turning the potential of a single cell into the wonder of a living, breathing being. Gently contemplate the poetic nature of this code itself. Unlike written language shaped by human hands, DNA's language is organic, shaped over billions of years. Its alphabet is a thread linking every breath you take with the origins of life itself. From the earliest, simplest organisms through every living thing, this same code persists, a testament to the continuity and shared heritage of all. Imagine for a moment the oak tree outside swaying gently as a breeze carries whispers of this story, the butterfly's delicate wings copying and adapting the same ancient letters, the tiny cells inside your own body renewing and recreating themselves through this tender blueprint. And so we settle enveloped in the quiet mystery and profound wonder of DNA. This delicate helix holding the infinite potential for life is both historian and architect, an ancient storyteller whispering softly through time. As the night deepens and you drift toward the gentle shores of sleep, carry with you the knowledge of this wondrous thread woven through all of existence, a reminder of the intricate, peaceful harmony that lies within and around you. Let your breath slow, steady and even, as if matching the quiet rhythm of DNA itself, the slow and purposeful unwinding and rewinding that keeps life unfolding gently, calmly, eternally, each cell turning its pages, reading its instructions, writing the ongoing story of being. Sleep now beneath the serene canopy of this ancient knowledge, cradled by the knowledge that your life too is written in this marvelous script, part of the endless continuity of life on earth. Rest in this quiet knowing as the story of DNA, the blueprint of life, wraps you softly in its embrace, guiding you toward dreams woven with the fabric of existence itself. And as the shadows deepen and the final echoes of the day fade, we will soon continue gently, exploring the wonders still held within this exquisite code. Until then, drift softly on the quiet currents of this ancient language, cradled by the spiral staircases of life itself. As the dim glow of our exploration lingers softly in the quiet corners of your mind, we now turn gently toward the next chapter of our tranquil tale. We have travelled in our thoughts through the vast, intricate world hidden within every living cell. We have touched upon the remarkable essence of heredity, that whispering legacy passed down through countless generations, woven invisibly into the very fabric of our being. Now we move slowly deeper into the heart of this silent library, where the stories are not just kept but lived, somewhere between the delicate strands of genes and the elegant structures called proteins, whose whispered instructions govern the dance of life. Imagine yourself nestled in the hushed aisles of an ancient vast library. The walls are lined with endless shelves, each holding the most fragile scrolls, precious with secrets. These scrolls are the genes, written in a mysterious script, a language composed of 4 letters, A, T, C, and G, arranged in exquisite sequences. Not unlike the careful arrangement of words and sentences, these sequences are the coded instructions, the blueprint of life as we know it. But our story is not merely about these scrolls themselves. It is about what happens when the library's wisdom breathes, when the knowledge is taken from written word into the realm of living action. This is the profound transition from gene to protein, a gentle transformation that animates the silent script with vibrant motion and purpose. Within our cells, these scrolls, our genes, tucked inside the nucleus, are kept under the soft, watchful care of molecular guardians. The story begins on a quiet day, without any sudden rush when a particular message from the genes is needed. Imagine a single scroll being carefully taken down from its shelf. This is the moment called transcription, where the language inscribed on DNA is delicately copied into a close cousin, a molecule known as messenger RNA or mRNA. Picture the process as if a skilled scribe copies the original text onto a special sheet, one that can leave the library of the nucleus to travel further. The letters on the DNA, the A, T, G, and C, are matched with their partners on the RNA. A pairs with U, uracil, replacing thymine. T with A, G with C, and so forth, like a carefully crafted shadow of the original. This copy, the mRNA, floats away softly, gliding out from the inner sanctum of the nucleus to the wider cytoplasm, the main hall of the cell. Here in the cytoplasm, the story unfolds further as the mRNA approaches the grand machinery known as the ribosome. If the nucleus is the quiet library, the ribosome might be thought of as a gentle artisan's workshop, where raw materials are brought together and shaped into functional forms. It is here where the magical process of translation begins, where the genetic code inscribed on the mRNA is read carefully and transformed into the language of proteins, sequences of amino acids arranged like beads on an ancient necklace. These amino acids, 20 in all, are the building blocks, simple yet profound. The ribosome reads the mRNA in triplet sets, the 3 letters together forming what is called a codon. Each codon is a word in the genetic language, specifying one particular amino acid. Slowly, the ribosome moves along the strand, reading each codon in turn, while helper molecules called transfer RNA, tRNA, bring the corresponding amino acid to the growing chain. Imagine this process as a serene, rhythmic weaving, a loom threading one strand after another, each thread a careful addition, precise and intentional. The protein begins as a simple string of amino acids, but it is destined for greater complexity. Silent molecules gently assist as the chain folds in upon itself, curling and bending into delicate shapes, sometimes like coils of mist, sometimes like intricate twisting ribbons, each shape essential to the protein's future role. Within these folds, something remarkable happens. The geometry of the protein determines its function, its ability to interact with other molecules, to catalyze chemical reactions, to carry oxygen, or to form the foundations of our cells. Some proteins become enzymes, softly accelerating life's myriad reactions. Others become structural, creating the scaffolding upon which cells build themselves. Still others serve as messengers and gatekeepers, transmitting signals and permitting the flow of ions and nutrients. It is a world of sublime precision and quiet purpose, where nothing is wasted, where the folded forms of proteins hold within them the wisdom of countless ages. And all of this, all the bustling life within, unfolds thanks to the poetic language encoded in our genes and to the gentle processing of the cell's intricate machinery. Pause here for a moment and let yourself drift into the gentle rhythm of this cellular story. Feel the slow passage of molecules moving softly like leaves on a calm stream. Each protein formed is a whisper of life, a promise of continuity, an echo from the ancient past, speaking in a language billions of years old. It is a language spoken not with sounds or words, but with the shapes and movements, the folds and bonds of tiny, incredible molecules. But proteins do not act alone. Many of them work in silent cooperation, forming vast, delicate assemblies. Imagine grand chandeliers of crystals suspended in a dark hall, each facet reflecting and refracting light. Similarly, proteins form complex machines, allowing cells to perform astonishing feats, from the repair of damaged DNA to the energy-producing workhouses that sustain every breath we take. Consider the mitochondrion, often called the powerhouse of the cell. Here, proteins are arranged into complexes that transform nutrients into usable energy. A process so mesmerizing it seems like a slow, glowing fire dancing beneath the surface. Without these protein assemblies, the cell would be as silent and still as a library without light, without warmth, without motion. Thus, a single gene's instruction, once transcribed and translated, can ripple outward in spreading waves of biological function, affecting everything from our ability to taste and smell, to our strength, to our very thoughts and memories. All comes back always to the exquisite dance of genes and proteins. Now, as we imagine this world deeper inside ourselves, behind the visible, beyond the skin, we also remember that this molecular symphony is exquisitely regulated. Not all genes are read all the time. Some remain folded away like secret manuscripts, only opening when needed. The cell listens attentively, responding to cues from itself and its environment, fine-tuning protein production with astonishing grace. Imagine now a candlelit room where the librarian moves quietly among the scrolls and tomes, opening some for a moment, closing others gently, guarding the balance of knowledge with tender care. Cellular regulators, hormones, and signaling pathways all play this role, delicately modulating the flow of instructions, ensuring harmony within the cell's ever-changing life. In times when the external environment shifts, perhaps the temperature changes or nutrients become scarce, the cell adapts. It might prioritize molecules essential for survival, curbing others until conditions improve. This dynamic responsiveness reminds us of the delicate balance all living things maintain, quietly and unfailingly. And yet, sometimes errors occur. Within this elaborate copying and translating, small mistakes can slip in. These mutations might be likened to tiny smudges on a scroll or a misread word. Most such errors wind up unnoticed or without consequence, as the cell possesses many wise proofreading and repair systems, gently correcting and smoothing the text. Occasionally a mistake persists, and the resulting protein can be altered, sometimes in ways that are benign, sometimes in ways that ripple into health and illness. Still, even through such faults, life carries on, with resilience born of billions of years of trial, error, and adaptation. As your thoughts wander further into this silent, hidden world, imagine the countless cells working in concert, each carrying its own set of genetic scripts, translating, folding, adjusting, responding. The harmony of this cellular community forms the basis of your body's form and function, the pulse of life flowing from this microscopic orchestra of proteins and nucleic acids. With eyes closed, you might envision these cells as tiny lanterns of life, each one illuminating a small sphere from within, glowing softly with the energy produced by proteins acting in unison. The light shifts gently, ebbing and flowing in tune with your own breath. And so, as your body grows more relaxed, your mind drifts like a leaf on a gentle breeze. Remember that within you lies a silent city of molecular artisans, tirelessly crafting, building, maintaining. The instructions within your cells hum quietly like an old beloved lullaby, a story told millennia ago by the ancestors whose genes found their way into yours. Listen now to this quiet song, gentle and unending, where every bend and fold, every amino acid, every transcription and translation moves softly and timely in. Each moment within your body is a testament to this marvelous secret of life, the language of genes, the artistry of proteins, the instructions within our cells. And as you settle deeper into sleep, know that these countless stories continue, silent and potent, a promise of regeneration, repair, and renewal, woven tightly into the night. Let yourself drift, carried by this ancient, unbroken wisdom. Rest now in the serene certainty of the molecular world nestled within you, a place of wonder, order, and gentle life. The library is always open, the scrolls always there, and in the darkness the proteins keep their steady pace, the guardians of life's quiet instructions of sleep. As the days and nights gently trace their eternal rhythm, and our minds begin to sink deeper into the quiet pools of rest, let us continue our slow journey within the unseen realms of life. Beyond the scaffold of bones and the fluttering pulse of blood vessels lies a world delicate and vast, a world where silent codes unfold and take shape in the softest ways. Today we will walk once more through the subtle, mysterious process of gene expression and regulation, feeling the slow breath of life as it whispers through the cells. Recall, if you will, our gentle introduction to the language written within the cells, the DNA, an ancient winding spiral holding an exquisite tapestry of instructions. These instructions, stretched out like tiny lines of poetry, do not simply lie silent. They move, they awaken, and they shape. This awakening, this transition from the silent script to something alive and tangible, we know as gene expression. Yesterday, with patience, we explored the basics, the transcription of DNA into RNA, the messenger that carries the thought from the depths of the nucleus outward toward the factories of the cell. Then the translation, where these messages become proteins, the humble builders and workers of life's grand design. But this evening, as your breathing settles into an even cadence and the world softens beyond your window, we turn now to the elaborate and tender dance of gene regulation, a symphony so subtle that it is almost invisible even to the eye. It is a dance carefully orchestrated, ensuring that the right genes speak only when needed, in the right amount at the right place and at the right time. This is no haphazard process, but one of profound elegance, one that can mean the difference between growth and quiet, between healing and stillness. Imagine, if you will, the cell as an ancient library, dim and vast, lined with shelves, not of books, but of these genetic scrolls, tightly wrapped, hidden within the shadows. The cell does not open every scroll at once. Instead, it has guardians and guides, molecules that roam these aisles with gentle hands, deciding which scroll to unfurl, which to leave tightly bound for now. These guardians, known as transcription factors, move quietly, like whispered voices recalling forgotten verses, coaxing the unwinding of certain pages only when their message is truly required. Before the scroll can be read, the gene expressed, the DNA itself must loosen its tight embrace. Picture the DNA as delicate twine wound around spools, histones, these spools, holding it secure, but also limiting its ability to reveal its secrets. The cell has learned to modulate this embrace, tightening or loosening as needed in a process called chromatin remodeling. Through subtle chemical touches, tiny groups added or removed, the histone spools either hug the DNA tightly, hiding the words, or relax their grip, allowing the cell's machinery to read the instruction clearly. This chemical ballet involves marks that feel almost like gentle letters written on the histones, acetyl groups, methyl groups, each changing the DNA's accessibility. Acetylation, the addition of acetyl groups, softens the grip, making the gene's message more available, like a candle flame glowing softly in the dark. Methylation, in contrast, often seals the script away, keeping it unread, a quiet guardian of silence. But even this is not rigid. Methyl groups can mean different things in different places, sometimes encouraging expression, sometimes silencing it, depending on the cell's needs. Within the nucleus, along with chromatin remodeling, there exist many other enigmatic elements called enhancers and silencers, sequences of DNA tucked away, sometimes far from the genes they regulate, yet connected through looping folds of DNA, much like the roots of ancient trees reaching deep, linking distant branches. They amplify or suppress the expression of genes, responding to signals from within and without the cell, ensuring the gene's message is finely tuned to the moment. But this is not the end of the tale. Even after the messenger RNA has been created, carrying the instructions out of the nucleus, the cell does not simply translate every message blindly. Here, in the fluid cytoplasm, further regulation awaits. The RNA itself can be modified, snipped and spliced in different ways, edited with precision, allowing one gene's message to create many different proteins, each with its own delicate variation, like different melodies emerging from a single song. This remarkable phenomenon, called alternative splicing, vastly expands the cell's ability to adapt and respond. Moreover, the lifespan of the messenger RNA is carefully controlled. Some messages are brief, a swift instruction that must be acted upon quickly, then discarded. Others are kept longer, as if waiting patiently for the proper moment to be translated into protein. Molecules called microRNAs and small interfering RNAs drift through the cytoplasm like gentle shadows, binding to messenger RNAs and guiding them to silence or degradation. They serve as silent moderators, ensuring that the cell's activity remains balanced and harmonious. Now imagine the cell's internal environment responding to the larger world, the flicker of light, the change of nutrients, the subtle effects of hormones. External signals reach the cell membrane like soft knocks on the door of this genetic library. Inside, they trigger cascades of molecular conversations, chemical messages passed from one protein to another, each activating or inhibiting the next, like a slow, echoing chain of whispered instructions. These signaling pathways ultimately influence gene expression, turning genes on or off in appropriate response to the environment. It is as though the genome listens attentively to the world outside and responds with a measured, thoughtful voice. Some genes are particularly sensitive to these external cues, the so-called inducible genes, lying quiet, waiting in their scrolls, only to come alive at the whisper of a signal. Others are constitutively expressed, forming the steady foundation of cellular life, like the walls of our library, always present, always guarding essential functions. This balance ensures survival and adaptability, growth and repair. The process of gene expression is also intimately woven with the cell's life cycle. As cells grow and prepare to divide, certain genes awaken to coordinate this grand event, while others recede into quietude. During development, from a single fertilized egg to a complex organism, the timing and regulation of gene expression is a masterpiece of precision. cells receive their identities, differentiate into varied types, each expressing distinct sets of genes, crafting muscles, nerves, blood, and bone. This regulation shapes the very form and function of life itself. Even in the quiet moments when cells rest or enter a dormant state, gene expression continues subtly, conserving energy while maintaining readiness for change. Epigenetic marks, these chemical memories on DNA and histones, remember past states and experiences, influencing gene activity without changing the underlying DNA sequence. This epigenetic regulation is like an ancient whisper, a soft echo from a cell's history, passed gently from one generation to the next. As we delve deeper, we see that regulation also exists on the level of the proteins themselves. Once synthesized, proteins can be activated or deactivated by the addition or removal of small chemical groups, by folding and unfolding, by fragmentation or relocation within the cell. Proteins that act as transcription factors, for instance, may lie dormant until a specific signal awakens them, then journey back to the nucleus to influence gene expression anew. The coordination of all these layers, chromatin accessibility, transcriptional control, RNA processing, RNA stability, translation efficiency, and protein modification creates a dynamic, responsive system. It is a living orchestra, playing softly and persistently, weaving the music of life, because without regulation, the cell would be overwhelmed by noise. It is this delicate control that brings harmony to the molecular world. Imagine in this stillness the countless cells across your body engaged in this ceaseless dance throughout the night. Each cell a universe of ticking clocks and whispered secrets, choosing which genes to express and when, in a choreography as old as life itself. Your breath, your heartbeat, the very pulse of your being rely on this subtle interplay, invisibly but inexorably. As your eyelids grow heavier and your mind begins to drift beyond the day's concerns, picture the genetic library softly closing its books for the night, quieting its guardians, returning to a gentle watchfulness. The DNA scrolls rest, wrapped neatly around their histone spools, while occasional flickers of light slide across their surfaces, memories of recent activity, promises of awakening to come. In the vast, soft expanse of your body's inner world, the process of gene expression and regulation continues, quietly sustaining life and guiding growth, healing and renewal. It is a process both ancient and ever new, a constant unfolding story written in silent ink, read and rewritten in harmony with every breath and every moment. Let this knowledge cradle you now, as you surrender to the deepening quiet, to the steady rhythm of sleep. You are a living symphony of molecules and cells, wrapped in an unending dance of expression and silence, balance and change. Let your thoughts drift lightly, your breathing deepen, your heart quiet, and let the ancient library of life settle softly into the stillness of night. And as you slowly drift away, carry the image of those tiny molecular guardians, those whispers of acetyl and methyl, those patient transcription factors and vigilant microRNAs, all working in concert to nurture and protect the delicate miracle of life within you. Sleep now, gently and deeply, cradled by the unseen dance that never ceases, the quiet song of gene expression and regulation. Rest well in the calm and steady presence of life's quiet breath. As we settle once more into this gentle journey through the realms of living things, the wondrous web of life encoded in delicate strands of molecules held within every cell, I invite you to breathe slowly, deeply. With each exhale, allow yourself to drift more lightly along the soft currents of this unfolding tale. In our earlier conversations, we ventured into the essence of genetic material itself, tracing the spiraling double helix of DNA, with its elegantly paired bases whispering across each rung. We imagined the graceful folding of chromatin, the symmetrical dance of chromosomes as they parted and reunited, carrying in them the sacred instructions that chart a creature's blueprints. We began to see how these threads of life are not inert symbols, but vibrant, dynamic guides, determining the potential of each cell, each tissue, each organ, the quiet architects of growth, change, and renewal. Now, as night deepens beyond the window pane, let us continue our journey inward, drawing closer still to the profound conversation between genes and life that unfolds within every living being. This, the story of how genetic information, so subtle, so small, shapes and directs the lifelong dance of development and the blossoming of traits. From the faintest flicker of a baby's first heartbeat to the complex tapestry of personality and form. Imagine the moment of conception as dawn breaking over a vast and fertile landscape. Within the union of two cells, the egg and the sperm, a new beginning stirs. Each brings a half complement of chromosomes, and together they restore the full symphony of genetic material. But this is only the first movement. The true opus begins as this single tiny cell reads its inherited instructions, waking ancient scripts embedded deep within a language of molecules and energy. From this nucleus, clutching its 46 chromosomes in humans, the cell begins to divide. One becomes two become four, and so the process continues gradually, gently, across days and weeks into a growing cluster of cells. At first, they appear similar, like identical notes in a quiet melody. But beneath this apparent uniformity, subtle shifts in genetic expression begin, some notes growing louder, some softer, guiding the destiny of each cell in ways as subtle as a whisper. These changes in expression are the cornerstone of development. DNA itself does not change, its sequence remains constant, but how it is read and interpreted varies. Like a well-loved book recited in many voices, different cells choose to emphasize certain sections, turning on some genes while silencing others. This selective reading, this intricate regulation, creates the diverse parts of a living organism. Think of this as an ancient forest where sunlight filters down through layers of leaves, casting patches of light and shadow. In some clearings, seedlings flourish and grow tall. In others, shade-loving plants thrive quietly beneath the canopy. The cell's environment within the embryo similarly influences gene expression, the biochemical signals, the mild pressures, the warmth or coolness, all contribute to shaping which genes awaken. During embryonic development, this finely tuned genetic conversation orchestrates the formation of tissues, skin, muscle, bone, and nerve, each with its own distinct pattern of gene activity. One can almost hear the soft rustling of gene networks like branches swaying, their connections growing stronger or weaker over time, guiding instructions to build the body's architecture. And so the organism takes form, its features reflecting not only the inherited genetic sequence, but also this ballet of gene expression. Curves of limbs, the gentle arch of a brow, the color of eyes, the density of hair, all appear as physical manifestations of genes given voice. Yet these visible traits are only a part of the story. Hidden beneath the surface lives a deeper complexity, the biochemical rhythms, the metabolic pathways, the faint electric hums of neural circuits, all subtly shaped by DNA's message. Beyond the blueprint of physical form, genetic information also influences deeper biological tendencies, how cells communicate, how they repair themselves, how they respond to the world around them. For instance, some genes teach cells the art of resilience, helping them withstand stress, illness, or injury, while others guide the timing of growth or rest. In this way, genetics forms an unseen hand, gently influencing the organism's inner symphony, its rhythm through time. Still, no creature walks its path alone. The expression of genes is not merely dictated by their sequence, but by a complex dialogue with the environment, both internal and external. This interplay is a delicate dance, where genes respond to signals, nutritional cues, temperature shifts, chemical messengers, even the touch of another living being. Consider the phenomenon of epigenetics, a term that hints at above genetics, a layer of information not written in the DNA code itself, but in chemical marks residing beside or upon the strands of DNA, like annotations in the margins of a manuscript. These marks act as switches or dimmers, modulating the reading of genes. Through epigenetic changes, life's experiences, stress, nourishment, warmth, can subtly alter how genes function, sometimes persisting through generations, like traces of distant melodies echoing down the corridors of time. This interplay reveals an exquisite truth about development. It is not a rigid unfolding of predetermined fate, but rather a responsive, adaptive process. Genetic information sets the framework, but the organism's journey integrates both inherited code and the rich context of its surroundings. Traits emerge not as fixed destinies, but as the result of an intricate balance, a harmony between script and stage, conductor and musicians. As the weeks and months pass in prenatal life, this gentle dialogue continues, shaping the neural pathways of the brain itself, the most complex organ of all. In these earliest moments, genetic instructions guide the formation of billions of neurons, each forming trillions of connections. Yet, these connections are not finalized by genes alone. Experience, even before birth, begins sculpting neural networks, movement, sound, warmth. The tender influence of the environment weaves alongside the coded plan. Through infancy and childhood, this conversation between genetic potential and environmental experience persists, growing ever more nuanced. Variations in DNA sequence, called genetic variants, may tip the scales subtly, affecting height, skin tone, metabolism, or predisposition towards certain health conditions. But these variations do not dictate with certainty, rather they nudge probabilities, creating a spectrum rather than a fixed outcome. Traits such as eye color or blood type follow relatively straightforward paths, often governed by a handful of genes. But many other characteristics, intelligence, temperament, susceptibility to diseases, emerge through complex interplays of numerous genes and countless external factors. Like a symphony performed by a vast orchestra where countless instruments and players contribute, the final music depends on subtle interactions and improvisations. This multi-layered process is why no two individuals, even identical twins sharing nearly identical DNA, are precisely alike. The minor differences in gene expression, influenced by environment, timing, chance, and even the microcosm of the body's internal chemistry, combine to create unique tapestries of traits and behaviors. In this light, genetic information is both intimate memoir and open score, steeped in history, yet receptive to new variations. As the story of development continues into adulthood, genes persist as custodians of maintenance and renewal. Each day, as cells age and wear, genetic programs activate repair systems, sending a quiet army of proteins to mend DNA damage, recycle cellular components, and maintain balance. Genetic influences extend beyond shape and form to subtle functioning, how our hearts beat steadily, how our immune system recognizes friend from foe, how we metabolize nutrients into energy. Even aging itself is deeply intertwined with genetics. Some sequences govern the length of telomeres, protective caps at the ends of chromosomes, which shorten with each cell division, marking the passage of biological time. Genetic variants influence how swiftly or gently this process unfolds, contributing to longevity and vitality. Yet just as development is shaped by environment and experience, so too is the aging process. Lifestyle choices, nutrition, activity, stress levels interact with genetic tendencies, influencing health and the manifestation of traits in subtle, unfolding patterns. This ongoing conversation between genes and life circumstances underlines the fluidity of our biological narratives. In this quiet reflection, it becomes clear that genetic information, for all its precision and complexity, paints a picture of life that is neither rigid nor predetermined. Instead, it reveals the profound beauty of biological potential, sculpted gently by chance, by surroundings, by relationships, and by time. Before we drift into deeper rest, imagine your own body as a library composed of countless volumes. Each cell houses the ancient tomes of your DNA, stacked with care on invisible shelves. Within these pages dwell the stories of your ancestors, the whispers of evolution, the intricate scripts that have guided life through eons. And yet each day new paragraphs are written by your choices, your sensations, by the quiet unfolding of experience that makes you uniquely you. So as you close your eyes and let the world soften into shadows and dreams, carry with you this understanding, a gentle, expansive knowledge that your being is a living manuscript, inscribed with codes by ancient forces, yet always written anew. Within you, genetic information whispers quietly, shaping the melodies of your form and spirit, guiding the endless symphony of growth, renewal, and transformation. Feel now how your breath becomes slow and steady, each exhale releasing the day's weight. The delicate conversation within your cells continues softly, unceasingly. Genes converse with molecules, molecules with cells, cells with tissues, weaving the fabric of your being, an endless, flowing dance, choreographed by life's most intimate instructions. Drift now, wrapped in the calm certainty of this inner story, as the night deepens around you. Let the subtle rhythms of genetic influence lull you toward rest, toward the soft edges of dream, where new growth and renewal await in quiet sanctuaries beyond waking time. Sleep comes gently, as does the endless mystery of life within you, encoded, evolving, embracing the night. As our exploration of dreaming in DNA drifts to a close, we can't help but marvel at the intricate dance of genes, whispering instructions like stardust across the cosmos of our bodies. I invite you to return as we continue to unravel more of these celestial mysteries tucked away in the quiet corners of our Dreamtime tales. Until then, may your dreams be filled with the gentle murmurs of your own genetic symphony. If sleep has already taken you, may your dreams stay gentle. If you're still awake, feel free to let your mind wander back over the quiet marvels we've uncovered. When morning comes, you can always return for another Dreamtime tale, and I'd love to know which mysteries you'd like to explore next. Sleep now among the gentle stars. Until then, thank you for sharing this calm corner of the cosmos with me. Rest well, breathe deep, and I'll meet you again beneath the stars. Good night.